We'll continue talking about cell structure and function with the discussion of the nucleus and the endomembrane system. The nucleus, as you know, is the command center of the cell. It's where the DNA is located, and it's where all the instructions for everything the cell has to do come from. The DNA is not recognizable as chromosomes except during cell division. The rest of the time it's more spread out in the nucleus, and we call it, when it's in that state, chromatin. The main purpose of the nucleus or the DNA is to direct the synthesis of proteins and other molecules in the cell. The nucleus is enclosed by a nuclear envelope, which is a double membrane, and its function is to control the flow of materials into and out of the nucleus. There are pores in the nuclear envelope that allow certain molecules to go through, both in and out, and others are kept out or kept in. Here we have a micrograph of the nucleus and a diagram showing you the different structures that we see here. The dark area in the center is called the nucleolus. There are pores or openings in the cell membrane. You see the pore structures here. The chromatin is just kind of a grayish looking or a more diffuse substance rather than the tightly wound chromosomes that we're accustomed to seeing. And then there are the endoplasmic reticulum with ribosomes located on the outside of the nuclear envelope. The nucleolus inside the nucleus is the place where ribosomal RNA is made. There are three main kinds of RNA we'll discuss later on, and one of them makes up ribosomes, which is where proteins are put together. The nucleus is in charge of directing the synthesis of proteins, and it does this by making a copy of the instructions called messenger RNA. These are the instructions for putting together a particular protein. The proteins are made at the ribosomes, which are found in the cytoplasm or attached to the rough endoplasmic reticulum. And the function of the ribosomes is to basically take the instructions from the messenger RNA and interpret them or translate them into a finished protein or polypeptide. Here we see ribosomes in a micrograph. You see these little brown dots here along the endoplasmic reticulum, and some just in the cytoplasm. And you find them all throughout the cell, and again, all cells have them. The ribosome has two subunits. It has a large subunit and a small subunit, and the messenger RNA kind of reads through there like a piece of tape, and that directs the putting together of the amino acids to make a particular protein. The endoplasmic reticulum is a factory where things are made, where molecules are made. It is a series of membranes that is involved in the synthesis and production of lots of different kinds of biomolecules. There are two kinds of endoplasmic reticulum. The smooth ER, endoplasmic reticulum, if we'll abbreviate ER, doesn't have any ribosomes attached to it. It is involved in making lipids, specifically, phospholipids for membranes and steroids for various other kinds of hormones and other molecules that are needed. In the liver, the smooth ER helps process harmful substances or detoxify harmful substances like drugs and alcohol. It also is involved in storing calcium ions that are necessary for nerve and muscle cells to work properly. Here we see the endoplasmic reticulum system. The rough endoplasmic reticulum is kind of continuous with the nuclear envelope there. It's connected to it, and then the smooth endoplasmic reticulum is a little bit farther away. The rough ER is more lamellar or kind of a series of layered membranes, whereas the smooth ER is more like a system or series of tubules. The rough ER is called rough because it has ribosomes attached to the surface, and it kind of looks like sandpaper. This part of the endoplasmic reticulum system makes proteins. These are proteins that are needed for membranes or organelles within the cell, or proteins that will be secreted by the cell for various purposes. And then these proteins are packaged into vesicles, which then can be processed by other organelles. The Golgi apparatus is also called the Golgi body or Golgi complex. It looks in the cell like a series of flattened sacks stacked on top of each other, or like a stack of pancakes. This is the warehouse and finishing factory for molecules that are made by the cell. There are enzymes in the Golgi that modify parts of the proteins called glycoproteins that have carbohydrate chains attached to them. It prepares any proteins for that are going to be exported from the cell for the export and packages them into vesicles, kind of like the shipping department of a factory or kind of like the UPS store. Here we see the Golgi apparatus in the cell, and you can see here the diagram. We've got a transport vesicle that has come from the ER, and it's entering the Golgi apparatus from this side, from the receiving side of the Golgi apparatus. And as the vesicles move through the stacks of membranes, the proteins that are contained in the vesicles are modified, packaged, folded, have parts attached to them. The quaternary structure of big proteins are put together and so forth. And as it moves through, it ends up on the shipping side, and it pinches off into a little vesicle here that can then move to the cell surface and be exported from the cell. This is a micrograph. An actual micrograph has been colorized, showing you the Golgi apparatus, the receiving side, and the shipping side here inside a cell. The endomembrane system is the term we give to these structures that are all involved together: the nuclear envelope, the ER, the Golgi, lysosomes, vacuoles, and plasma membrane. And they all work together to take care of the molecular needs and requirements of the cell. And they're also involved in separating the cell into separate compartments. This is important for the different reactions that go on in the cell. They may require different kinds of conditions in order to function properly. The lysosomes are organelles that contain digestive enzymes, and their function is to recycle things within the cell, to digest ingested food particles, and so forth. Vacuoles are storage compartments that store either food or water or other kinds of molecules for later use. In plant cells, like we saw before, there is a large central vacuole that's used to store things that are made by the cell. Some single-celled organisms have these specialized vacuoles called contractile vacuoles that get excess water and expel it from the cell. Here we see lysosomes, a lysosome fusing with a vesicle containing something that's been damaged in the cell to digest or break it down. Here we have a plant cell with its central vacuole there, showing that this one contains starch, it appears to. And then here is a paramecium with its contractile vacuoles that are used to get rid of excess water. Peroxisomes are another kind of organelle that is found in most cells. These have enzymes that are in the form of a crystal. They're oxidases that can break down harmful chemicals such as alcohols to hydrogen peroxide, and then into water and oxygen. Hydrogen peroxide, as we already know, is harmful to cells, and so you have to have a way to deal with it because it definitely is a breakdown product of a lot of other molecules. So lastly, we just want to see the connections between all these things. This is why it's called the endomembrane system, because you've got a system of membranes surrounding the nucleus and making up the ER, and also making up the Golgi apparatus, and then the lysosomes as well are made of membranes that come from these sources.